Photosynthesis is the process by which cells synthesize organic molecules from inorganic molecules in the process of sunlight. Photosynthesis is done in two steps: the light-dependent reaction, which converts light energy from the sun into chemical energy, and the light-independent reaction, which uses chemical energy to photosynthesize organic compounds. The light-dependent reaction is made up of three steps. Step 1 is the excitation of photosystems by light energy. The photosystems are groups of photosynthetic pigments that are embedded within the thylakoid membrane. Photosystems are classified according to their maximum absorption wavepoints. When a photosystem absorbs light energy, delocalized electrons within the pigments become excited or energized. These excited electrons are transferred to carrier molecules within the thylakoid membrane. Step 2 is the production of ATP via an electron transport chain. Excited electrons from photosystem 2 are transferred to an electron transport chain within the thylakoid membrane. As the electrons are passed through the chain, they lose energy, which is used to translocate H+ ions into the thylakoid. The buildup of protons within the thylakoid creates an electrochemical gradient, or proton move force. The H+ ions return to the stroma through the proton gradient via the transmembrane enzyme ATP synthase, or chemiosis. ATP synthase uses the passage of H+ ions to catalyze the synthesis of ATP from ADP plus P. This process is called photophosphorylation, as light provided the initial energy source for the ATP production. The newly de-energized electrons from PS2 photosystem 2 are taken up by photosystem 1. Step 3 is the reduction of NADP plus in the photolysis of water. Excited electrons from photosystem 1 may be transferred to a carrier molecule and used to reduce NAPH plus. This performs NADPH, which is needed for the light-independent reactions. The electrons lost from photosystem 1 are replaced by the de-energized electrons from photosystem 2. The electrons lost from photosystem 2 are replaced by electrons released from water via photolysis. Water is split by light energy into H+ ions and oxygen. The light-independent reaction, also known as the Calvin cycle, takes place in the stroma and also has three steps. Step 1 is carbon fixation. The Calvin cycle begins with a 5-carbon compound called ribulose biphosphate, or RUBP. Ribisco, or RUPB carbolysase, catalyzes the attachment of a CO2 molecule to RUBP. That results in a 6-carbon compound that is unstable and breaks down into two 3-carbon compounds called glycerate 3-phosphate, or GP3. A single cycle involves 3 molecules of RUBP combining with 3 molecules of CO2 to make 6 molecules of glycerate triphosphate. Step 2 is the reduction of glycerate triphosphate. Glycerate triphosphate is converted into tyrose phosphate using NADPH and ATP. Reduction of NADPH transfers hydrogen atoms to the compound, while the hydrolysis of ATP provides energy. Each G3P provides one NADPH and one ATP to form a tyrose phosphate, so a single cycle requires 6 of each molecule. Step 3 is the regeneration of RUBP. Of the 6 molecules of TP produced per cell, 1 TP molecule may be used to form half a sugar molecule. Hence, 2 cycles are required to produce a single glucose monomer, and move to produce polysaccharides like starch. The remaining 5 TP molecules are recombined to regenerate stacks of RUBP. The regeneration of RUBP requires energy derived from the hydrolysis of water.